Hi, we're going to learn about bacteriophages in this presentation. So, like I said, bacteriophages are special classes of virus which are exclusively inhabiting on bacteria for their survival needs. So, being obligate intracellular parasites, virus will always look for a suitable host to continue with their multiplication and to produce plenty of offsprings to continue with their generation. And this class of virus, I'm talking about bacteriophages, they will eat bacteria. That's why the term bacteriophages. Phages means eating. Bacterio means something related to bacteria. So bacteriophages are, they are not bacteria, they are virus which are inhabiting bacteria for their survival needs. So let's go to the presentation and we'll see the basic characteristic features of bacteriophages and their applications in clinical microbiology. So these are virus which are infecting bacteria. That's a basic definition, and they are readily isolated in fecal samples, sewages, and natural sources. That means it's present everywhere. So these classes of virus are present everywhere, and even if it is surviving in human body, it's not dangerous to us to have a bacteriophage in our body because they won't cause multiplication or they won't cause any kind of complications in our own cells, but they can damage a bacterial cell, and they are the ideal models for virus studies. And we have taken plenty of bacteriophage cultures because these are something which can be easily isolated and easily grown in lab condition. Because if I have to grow them in lab condition, the best culture medium will be having a bunch of bacterial population. That's it, because they are something which is growing in bacterial population. So it's easy to grow them in lab conditions. So I can study more on viruses by focusing on these these bacteriophages. They can be grown easily on bacterial cultures. So the significance of bacteriophages includes, these are agents which can be used for genetic transmission of characters from one generation to another generation. We have learned about transduction in the general microbiology, where we learned that transduction is a process where there is genetic transfer happens between two bacteria, and that is happening through bacteriophages, and that is coming under genetic transmission. And through genetic transmission and transduction, there will be some slight changes in bacterial characters, and that is coming under the heading phage conversion. So we have genetic transmission, and which is followed by phage conversion, which will end up in phage conversion. Phage conversion means change in bacterial characters as a result of involvement with the so change in bacterial characters as a result of the involvement of bacteriophages over their population. So with this, we can make some manipulations or genetic manipulations in the bacterial family. And this is something which we used in genetic engineering, where we have to produce plenty of one simple example is introducing insulin genes into bacterial cells. We can use bacteriophages. They can act as vectors for transmission of a particular gene from one species to another species. And the same bacteriophages are also used to control bacterial population, because these are agents which are targeting bacterial cells. I hope you have heard about superbugs, right? Superbugs are those, these are kind of specialized or modified forms of bacteriophages which can damage wide variety of bacterial populations. So we can, for example, if there is a water source and if you suspect severe bacterial contamination in it, what is the best thing we can do is that we'll introduce some kind of superbugs or bacteriophages into there, and these superbugs will destroy or kill the bacterial population over there. And the same bacteriophages can also be used to type the bacteria. Typing means identification of a bacterial species in lab condition. That can also be done using bacteriophages. So for general study purpose, we have taken TE1 phages, which is infecting E. coli, and we'll see their morphology and special characteristic features in this presentation. So they are tadpole-shaped bacteriophages, which has got an hexagonal head with a double-stranded DNA as genetic material, with a cylindrical sail and hollow core, contractile sheath, terminal base plate with prongs or tail fibers. So let's see the actual image of uh, tadpole shape TE1 phages, which is something which is infecting E. coli. So they got icosahedral or cubical symmetrical head, which is having double stranded DNA as the core, and this has got a tail core with a contractile sheet, which is helping them to, some kind of flexibility, which ends with a base plate with some plenty of tail fibers, and this is helping them to attach with the host cell. And here the host cell will be E. coli. Bacteriophages, the bacteriophages here we have taken TE1 phages. They are inhabiting on E. coli. Right. So if you see the life cycle, they follow almost similar steps of a normal virus in terms of the replication. There will be adsorption, penetration, uncoating, biosynthesis, assembly, and release. But at the time of release, it can opt two options. Either they'll end up with a lytic cycle or lysogenic cycle. So lytic cycle will be more virulent. This is almost same like normal viral replication. Lytic cycle will be more virulent, where what happens is the newly formed bacteriophages will break open the host bacterium and will come in large numbers to the surrounding environment. That will be really dangerous to the E. coli. That's why we have included that as a virulent cycle. Right. So the second option will be, the second possibility will be a temperate cycle or a lysogenic cycle, where the bacteriophage will not come out in large numbers by breaking the host cell. Instead, they will introduce their double-stranded DNA part to the host cell genetic complex, and they'll wait there for a right opportunity. So DNA phages gets integrated into the bacterial genome, replicates within with it causing no harm to it. That's a temperate or lysogenic cycle. So you can make out in this image I have mentioned both lytic cycle and lysogenic cycle. So here you can see here there is entry of a bacteriophage, tadpole shaped bacteriophages. They'll attach with the host cell surface by adsorption. They'll penetrate the genetic material into the cytoplasm, and these nuclear material will continue with the lytic process, lytic cycle. They will produce plenty of new viral components and viral DNA materials, and they will undergo replication. And once they achieve maximum number of all the components of the virus, they will undergo packing process, and these packaged items will burst out by lysing the host cell. They'll come in large numbers. This is the lytic cycle. But when you talk about lysogenic cycle, here there is no lysis happens for the host cell. Instead, the bacteriophage DNA is getting incorporated with the host cell nuclear material. That's it. There is no further cell division happens. So with this, in lysogenic cycle, the bacteria is getting a new character. The host cell is getting a new character here. New character here. There's no damage to the host cell, whereas in bacteriophagic lytic cycle, complete damage happens to the host cell, and large number of bacteria will come to the surrounding environment. So these steps we have already discussed. They follow absorption. Adsorption is actually not adsorption. They'll undergo penetration, synthesis. So let me change it for you. Alright, so there will be adsorption, and where that's during the absorption, bacteriophage tail fibers will interact with the specific receptors on the host cell body, and followed by penetration, where they will introduce their nuclear material into the host body, followed by the synthesis of phage components, early proteins and late proteins, same like normal viral replication. So you can see the viral particles, the bacteriophage particles are interacting with the host cell surface. This is adsorption, and intimate in contact. This fibrils will bring the bacteriophage main body, which is very to very close to the host cell body, and followed by penetration. During the penetration process, the nuclear material of the bacteriophage will be introduced into the bacterial body. And next will be the synthesis of new viral particles, assembly of bacteriophages, and finally everything will come in large numbers. So in virulent or lytic cycle, assembly will happen. There will be maturation, release of progeny phage particles, and can be plotted in on one-step growth. It actually happens like in the format of a growth curve, where large number of new viral particles will escape. Progeny virus will escape into the surrounding environment by breaking in host body. So we have to learn about few more technical terms. Eclipse phase. Eclipse phase is the time interval between the entry of the phage nucleic acid and the appearance of cell infectious phage particles inside the cell. So here eclipse phase is the eclipse phase is the phase or the time gap between the entry of viral particles and the appearance of new viral particles inside the host cell. Whereas in latent period, it's a time interval between infection of the host cells and increase in extracellular virus. So here, this is the time gap between entry of virus and the appearance of new viral particles outside the host cell. These are two terms which we have to learn, which we have to memorize in this slide. So, in case of lysogenic or temperate cycle, phage DNA integrate with the bacterial genome. Prophage replicates synchronously without harming the host cell, so the bacteria is getting a new character, right? So, we can employ this technique to bring new characters to the bacterial cells. Now, significance of bacteriophages. So, virulent phages can be used for phage typing, identification. So, what we can do is that, for example, if you got a mixture of bacterial colonies on a culture plate and we don't know which is it, and what we can do is that we'll use some random bacteriophages over it, and we will place bacteriophages in a random location, and we'll locate like we will note out, we'll make a note like which bacteriophage I have given to which location, and I'll subject this for incubation. And after incubation, if there is specific damage to those areas, we can have an idea about the bacterial population which is present on the culture plate. So, in case of temperate phages, we can use it for phage conversion and transduction to bring new characters to the host cell. Yeah, this is actually phage typing. So here, what they have done is they have inoculated the bacterial species on this culture plate, and in random location they have introduced the bacteriophages, and they know like in this location which bacteriophage I have introduced, and here which bacteriophage, and here which bacteriophage. So they'll make a note of the list of bacteriophages which they're going to implant on this culture plate, and this entire plate will be incubated. And those areas where there is no growth, for example, in this area there's no growth, that means the bacteriophage which I have introduced here can kill the bacteria. So that means the bacteriophage shows some kind of specificity to this kind of bacteria, so I can have an idea about the underlying bacterial population. Right, so that's all about bacteriophages. Bacteriophages doesn't have any clinical importance, but it's mainly used for diagnostic and research purposes.